In this podcast, we're going to go over what osmosis is, what dialysis is. We'll also talk about what happens to cells when they're placed in solutions with different concentrations of solute, and we'll do some application of dialysis to the nursing profession. All right, so before we talk about osmosis, let's talk about this idea of a semi-permeable membrane. So semi-permeable membrane, which is what you're seeing here in gray, will allow small solutes and water to pass through freely, but not large molecules. And in the case of this picture, we see our solute is a pretty large molecule. It will not fit through there. So only water, which is depicted in this image, is going to be able to pass through the semi-permeable membrane. When we talk about osmosis, we're talking about the flow or the movement of water. So water is going to move from a side of a semi-permeable membrane with a low concentration of solute. That's an important word, low concentration of solute, to the side of the semi-permeable membrane that has a high concentration of a solute. So the movement here is from low to high solute. So in this image, we see that there is no solute on the right side of your image, but there is solute and solvent. So this is going to have more solute. It's the higher solute. So we're going to see water flow from the right to the left of the semi-permeable membrane. It's going to flow from low concentration of solute to high concentration of solute. The higher concentrated solution, because we're adding a bunch of water, we're not changing the amount of solute, we're just adding more water. It's going to become more dilute as water moves in. All right, so this image is a lot better for me for talking about the movement of water osmosis because it shows us what actually happens to the level of water after you have movement. So real quick, let's talk through the image and we'll go back to osmotic pressure. So here we have a side that has water only, and we have another side that has a solute or glucose dissolved in water. So because we see one side that has a lot of solute, a high concentration, and water is a low concentration or a no concentration, we're going to see movement from the side of low concentration of solute to the side of high concentration. Well, over time, what happens is you have one side filling with water. The attempt is to dilute that side enough so that you have equal concentrations on both sides of your semi-permeable membrane. This isn't going to happen because there's no solute on the left-hand side, but the attempt is to dilute that side. So what happens is you get something called osmotic pressure. So we have the movement of water to one side of the membrane. And osmotic pressure, at some point, as you keep adding water across this membrane, you're going to get an increased pressure on that membrane. Eventually, that back pressure will prevent the flow of additional solvent into a solution on one side of the membrane. So at this point, we can call this the end point. You can no longer get any more water over that boundary, over that semi-permeable membrane. You have reached the maximum amount that it can go. Now, this is a colligative property. It does depend on the number of particles in a solution. We're going to talk about colligative properties, not in this podcast, but actually in class. The more particles that I dissolve into this water, the greater the osmotic pressure you're going to get. And that has to do with the fact that you're going to have to pump more water over to try to dilute that solution. Now keep in mind, this one's kind of tricky, is once you have reached your maximum amount of water, that doesn't mean that water stops flowing. Okay, you cannot go any higher in your amount of water on here, but what happens is you get equal movement of water back and forth across that membrane. It never just shuts down. Okay, you'll get some molecules in, other molecules out across that membrane. The flow does not stop, it just stops increasing the amount of water on one side of the semi-permeable membrane. Some terms I want you to familiarize yourself with describe the solute concentration on either side of a cell. So the first is an isotonic solution. So let's just use a random cell, and we will use solute concentrations inside the cell as dots and outside of the cell. I only use 3 particles of solute because it's easy. In an isotonic solution, the amount of solute particles inside the cell is equal to the amount of solute particles outside of the cell. So what you're going to get is equal flow of water into and out of. We're not trying to push water to one side to dilute it so the concentrations are equal. So equal flow in and out, they have the same osmotic pressure. In a hypertonic solution, you are going to see the solution outside of the cell, hypertonic, is more concentrated. So here we'll just put our 6 over here and maybe just 1 inside. So I have less solute inside the cell. Well, as you know, with osmosis, it's the movement of water from a low concentration to a high concentration. So the water is going to rush out of the cell. And the goal here is to dilute that concentration outside of the cell. And it would like to dilute that concentration. We see this in red blood cells. If you were to place them into a salt solution, that salt solution is more concentrated. So your red blood cell is actually going to shrink as the water moves out of the red blood cell. And we call this process crenation. So, and if this is going to be a terrible drawing, I have a great image on the next slide, so hang in there. But it will eventually shrink as all the water has moved out of the cell because the volume is shrinking because those red blood cells have a lot of water in them. In a hypotonic solution, we've got the opposite situation going on. The solution outside of your cell is a lot less concentrated than the solution inside your cell. So you have a higher concentration inside the cell. Again, in osmosis, you have movement of water from a low concentration to a high concentration. So water is going to rush into the cell to try to dilute the solute concentration inside the cell. If I were to take the same red blood cell, stick it in pure water, like distilled water or DI water that you use in lab, it's a lot less concentrated outside the cell than inside. So what happens is your red blood cell starts to swell. Well, eventually you're going to take on so much water, your red blood cell is going to burst. This process or this situation is called hemolysis, where you have bursting or opening, splitting of your red blood cell. All right, so their pictures are a little better than mine, but essentially here's what's happening. You have an isotonic solution and you have equal amounts of solute inside the cell and outside of the cell. So you're going to get equal movements of water into and out of the cell. Remember that flow doesn't stop just because it's isotonic, and iso means the same. So you get equal flow into and out of, and you get these beautiful disc-shaped red blood cells with a little depression on there, great surface area, very good for picking up oxygen. When you put these into a hypotonic solution, you don't have very much solute outside, if any, but you have a ton of solute inside. So what happens is all the water rushes into the cell, and eventually it could potentially burst if you get too much water running in. So now if you look at some of these, this isn't a great image, but you see that they start to kind of bubble up into a bigger, rounder shape. They're not that nice flat disc anymore. Eventually that's going to burst because your cell membrane wasn't meant to deal with that osmotic pressure created by all that water rushing in trying to dilute your internal solute concentration. And finally, in our last situation, you have, say, a salt solution with lots of concentration of solute outside. The water is rushing out, and so the cell essentially shrivels up. It loses its water to the outside, external, extracellular environment, and your cells are going to start to kind of shrink, and they take on these weird-looking, shriveled up, crenated red blood cell. In dialysis, we're separating solution particles from colloids. This is going to, a dialysis membrane is going to allow water and small molecules as well as ions to pass through a semi-permeable membrane. This is a very selective process, and in your body we see this in the kidneys. So your kidneys have millions of nephrons, which are tube-like structures. They have filtration membranes in them, and so as blood passes through there, we're able to filter out small molecules such as glucose, amino acids, ions, urea, water, and we can either reabsorb the useful materials such as water or glucose or amino acids, things that our body can still use, but we can also filter out those waste products like urea, and then those can be eliminated from the body. But this does use the same idea, this dialysis, this semi-permeable membrane where some particles can pass through and others cannot. And it's a way for us to clean the waste products out of our blood without losing all of those important nutrients. A great application of dialysis and this idea of the selectively permeable membrane is when a person is incapable, their kidneys no longer function to remove waste from the blood, we put them on hemodialysis or dialysis treatments. So what essentially happens is you have an individual's blood flowing through this machine, and within this machine you have a cellophane membrane inside, and it is suspended in an isotonic solution that has sodium chloride, potassium chloride, sodium bicarbonate, as well as glucose in there. And it's able to filter the urea and the waste products out of the blood through the semi-permeable membrane because the kidneys are no longer able to do that. However, because it's a semi-permeable membrane and only small solutes can pass through, it's not able to remove blood cells from that or other large molecules. Those are just too large for the machine or for the semi-permeable membrane. So it's able to keep function the same as the kidneys in that it can keep all of those nutrients and all of those recyclable goods for the body, so you're not having to constantly make extra blood cells when you can just reuse the ones you have. Typically, if an individual is on dialysis treatment, it does take a couple hours for this process for all the blood to be filtered through the dialyzer, and you would need about 2 to 3 treatments per week depending on the functioning of your kidneys at that particular time.